Hello and welcome. In this session, I'm going to discuss about two aspects of bacterial plasmids. That is, how plasmids maintain their number in a cell generation after generation. And secondly, what is plasmid incompatibility? Now, before I come to these topics, I would like to refresh your memories about some facts, what we know about plasmids in general. So, as you may recall, plasmids are circular and self-replicating extragenetic material present in prokaryotic cells or bacterial cells. Now, plasmids per se are not required or necessary for bacterial cell growth, but they do have certain genes which are useful for bacteria or prokaryotes, like antibiotic resistance, fertility, you know. So not only prokaryotes, some of the lower eukaryotes also have been shown to possess plasmids. I'm not going to talk about this aspect, but let us continue about prokaryotic plasmids. Now, as far as their size is concerned, it varies from few kbs to few hundred kbs. And plasmid replication, as you may recall, it uses its own origin of DNA replication, but it uses the host cell DNA replication machinery to complete the process of replication. Except some recombinant plasmids, which have their own uh, replicating enzyme on it. Other than that, other than that, this structure, what we call plasmids, as you may recall, is the basis of modern day recombinant DNA technology. So, with this kind of background, let us now address the question of today, that is, how plasmids control their number or copy number in a bacterial cell. Okay. So, before I proceed further, let me tell you that one of the ways to classify different plasmids is based on their number in a cell. If a plasmid is present in less numbers or lower numbers, say 1 to 9 or 1 to 10, it is called as stringent plasmid or low copy number plasmid. And if a plasmid maintains a high number, that is 10 and above, we call that as relaxed plasmid or high copy number plasmid. So this is one way of putting different plasmids into these two categories based on their number present in a cell. Now, the number of these plasmids in a cell, as we know today, is regulated by controlling DNA synthesis. Okay, so DNA replication is the key for maintaining a particular number for a plasmid in a cell. Keep this in mind. Okay, so basically the it is the control or the regulation of the plasmid replication or plasmid DNA replication, which is responsible for the, which is responsible for maintaining a particular plasmid number in a cell. Now, before I address to this question, you know, a basic question was asked that whether it is the host cell or the plasmid itself controls the number, control its number. Okay, so a very simple experiment was performed, which answered this question. And the experiment goes like this. You know, a plasmid free culture was maintained, established and maintained. And then this culture was on one hand transformed with a low copy number plasmid, and on the other hand, the same culture was transformed with a high copy number plasmid. And the transformation was such that each cell receives not more than single plasmid in a cell, here as well as here. Now, after these cells were allowed post-transformation in both the cases, when the plasmid number was checked, you know, so when a batch of these cells was transformed with a low copy number plasmid, here the plasmid replicates once or twice and then stops. But this batch of cell which was transformed with a high copy number plasmid, here the plasmid continues to replicate until it reaches to a number which basically it maintains in a cell. Okay, so what we are seeing here, that the host cell is the same, but when it is transformed with a low copy number, the plasmid maintains a low copy number here. And when the same cell is transformed with a high copy number plasmid, the plasmid maintains high copy number. So what is the conclusion? Conclusion is that the host cell basically has nothing to do with controlling the plasmid numbers in the cell. Okay, so it means it is the plasmid which decides that whether it is going to maintain a high copy number or a low copy number. Means if a low copy number plasmid enters into the cell, it maintains the low copy number. But if a high copy number plasmid enters in the cell, then it maintains the high copy number. Okay, so simple experiment like this gave an idea that host cell has nothing to do with maintaining the copy number, but it is the plasmid itself which decides that how many uh, copies it is going to produce. Okay. Now, if that is the case, let us see how then plasmids control this feature. So, a mechanism was proposed, okay, and it was hypothesized that plasmid encode for a repressor, now repressor for replication, repressor which regulates the initiation of replication, and that too in a negative form. Means so long the repressor is there, it will not allow plasmid to replicate. So this repressor is negatively regulating the initiation of replication. So that is the hypothesis made. And also another assumption was made that the activity of repressor, it depends on its concentration in a cell. So a particular concentration is required to keep keep the plasmid replication in check. Means if the concentration of repressor goes down, it allows the replication to go on. But when the repressor concentration increases, then it stops the plasmid replication. So these were the two assumptions or hypotheses were made that the plasmid number or plasmid regulates its replication via a repressor, which is negatively regulating initiation of replication, and this is dependent on the concentration of the repressor. Fine. Now, how this then mechanism works? So what happens for any cell to divide, whether it is prokaryotic or eukaryotic, first thing, that cell increases in size. The cell size increases, and then the cell components also doubles, whether it is the DNA amount or the other cellular components also increases. And then because the cell is preparing for itself to divide into two. So when the cell is growing in size, what is happening? The 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 repressor concentration goes down. And when the repressor concentration goes down, replication is not, the plasmid replication is not inhibited, and the number of plasmid DNA doubles. Means it gets, after replication, plasmid replication, one plasmid becomes two plasmid. Now, when the two plasmids are generated, now there are two copies of that repressor, which is two copies of this repressor gene, which is making the repressor. So the concentration in the cell again increases and doubles up. And then the replication stops. So basically, what's happening here, that with the increase in the cell size, the concentration of the repressor drops, which allows replication to occur, and the plasmid gets doubled, or the one plasmid becomes two plasmid. And then when there are two plasmids in the cell, they both are making repressor, and therefore the concentration goes up again, the concentration of the repressor, and it stops the replication. Now, this hypothesis or this explanation explains both high copy number plasmid behavior as well as the low copy number plasmid behavior, in the sense that a high copy number plasmid will have high concentration of repressor, and a low copy number plasmid will have low concentration of repressor. Okay, so it means, what I'm trying to say is, if there are two plasmids in a cell, and when the cell is dividing into two, the plasmid also should become 4, 2 to 4, so that each daughter cell may have or should have two plasmids. Okay, so when this cell containing two plasmids is preparing itself to divide, it grows in size. So the repressor concentration goes down, and the DNA replication takes place, and the two plasmids become 4 plasmids. And these 4 plasmids then make repressor, and when the concentration increases, it shuts the plasmid replication. Similarly, if it is a high copy number plasmid having 20 plasmids in the cell, so 20 plasmids are making repressor and keeps the plasmid replication in check. But when this cell prepares itself to divide, it increases in size, and the repressor concentration goes down, so that all 20 plasmids, they duplicate themselves, they become 40. And the 40 plasmids, when they make repressor, again the repressor concentration goes up, and it shuts the plasmid replication. So that's how this mechanism, which was proposed and explained, very clearly explains the behavior of both low copy number as well as high copy number plasmids. Now let us look at some specific experimentation. So please follow this experiment with me very carefully. So we have a plasmid we call E1. And we have another plasmid, that is PSE101. This is a recombinant plasmid, and this is the natural plasmid occurring in bacterial cells. Okay. Now, in a normal cell, wild type cell, which is Pol A plus, this plasmid maintains 16 copies. Okay, and it's a high copy number plasmid. And PSE101, it maintains 6 copies in a cell. So it's a low copy number plasmid. Let me tell you here that col-E1 uses host cell DNA polymerase for replication, for its replication, whereas PSE101 has its own replicase so that it doesn't depend on the host DNA polymerase. Now, when these plasmids are in a mutant for Pol A minus or Pol A minus cells, then what happens? See, col E1, which in a normal cell maintains 16 copies, it's not able to replicate. So one copy remains one copy. Whereas PSE101, it is able to replicate, as I mentioned, because it doesn't require Pol A, the host cell Pol A. So it's able to maintain its own copies here, which is 6. Okay, so this is the behavior of these plasmids in a normal cell and in a mutant cell. Fine. Now, what was done here, that these two plasmids were fused, okay, to get another recombinant plasmid that was named as PSC134. Okay, now this recombinant plasmid, it has two DNA replication origin, one of called E1 and another coming from PSE101. So it has both the origin of DNA replication, whereas these have only one for their replication. Okay. Now, when this recombinant plasmid or fused plasmid, you can say, was used for transformation and put it in a cell, in a normal cell and in a mutant cell, mutant for A or A minus. So let us see what's going on here. So when this fused plasmid was moved in a normal cell, so it is utilizing the replication, DNA replication, origin of DNA replication of this plasmid, that is called E1, which is a high copy number plasmid, and it requires a higher amount of repressor to inhibit its replication. So this replication continues, and you see the 16 copies of this fused plasmid in a cell. Okay, in a cell. So you have 16 copies. Now, when it moves to the mutant, which is A minus mutant, so plasmid replication doesn't take off here, but instead it takes off from the PSC 101 origin of replication it uses. And this is controlled by lower amount of repressor, so that, it only makes 6 copies. Because the origin of plasmid replication is used here is from PSC 101. Note this one, because Pol A is not there, which takes off. So it maintains only 6 copies. So the point I'm trying to make here, that although the fused plasmid has both origin of replications for a high copy or a low copy, and depending on the cell where it goes, if it is going into the normal cell, then a high copy number of this plasmid is maintained. And if it goes into a mutant, allowing the other plasmid's origin of replication to be utilized, then it maintains the lower copy, that is 6 copies. So what one is seeing here, that the replication or the plasmid number is dependent on the replication of plasmid, how many times it is going to replicate or how many times it is allowed to replicate. Okay, in one case, it is allowed to replicate 16 times. Okay, and in the other, it is allowed to replicate only 6 times. So basically, it is this property of plasmids, which they exploit to maintain their number in a cell generation after generation. You know, this was further tested and confirmed, means this concept of high repressor amount, low repressor amount, regulating the replication. You see, in this experiment, what was done in the same cell, which has these fused plasmid, PSC134, here it has 16 copies of this, a very high level of repressor in this cell. Now, when this cell was transformed with PSC101, you know, okay, in such a way that single copy of plasmid goes inside. Now, the expectation is that since this maintains 6 copies, it should be able to replicate and make 6 copies here. But what was seen both in normal cell or in mutant cell, this plasmid doesn't replicate at all. Why? Because this already has 16 copies of the plasmid, which is making very high level of replication repressor, which we are considering. And because of that, this plasmid is not able to replicate. So it further confirms that the plasmid coded repressor is the key which regulates the replication of plasmid, you know. So experiment like this clearly demonstrated that that the copy number of a plasmid is maintained by itself through a repressor, which negatively regulates the replication of plasmid. Okay, and an appropriate concentration is required. This inhibition of plasmid replication is concentration dependent. Okay, now let us come to the second part or second question of today, that is plasmid incompatibility. You know, so what is seen actually that there are some plasmid types which can coexist in a cell. We call them as compatible plasmids. But there are other plasmids which cannot coexist in a cell or cannot be maintained in a cell, if you want to say it that way. So that we call it that these plasmids are incompatible. Okay, so the incompatibility of the plasmids can also be explained by this repressor model, which I explained to you earlier with respect to maintaining the copy number. The same repressor model can I also explain the plasmid incompatibility, that why two plasmids cannot maintain themselves together, you know. Now, for example, if you have these two plasmids, F plasmid and called E1 plasmid, you know. So there is a cell, host cell, which contain these two plasmids. And these two plasmids are maintained generation after generation together. Okay. So what is happening here? These two plasmids have the repressors which are different. Means F plasmid has its own repressor and the col-E1 has its own repressor of plasmid replication, you know, and they are not interfering with each other. So therefore, these plasmids, they replicate independently to each other and maintain their copies in a cell without interference from each other. So it means the repressor of, say, F plasmid does not regulate the replication of col-E1 and vice versa. Means these repressors are independent and they are not interfering with the replication of the other plasmid. And therefore, these two types of plasmid can maintain themselves independently. But what happens when you have two plasmids which share this repressor or whose repressor is similar or identical? Then the problem comes. It means suppose you have two cells A and B who are sharing the repressor. Repressor of what? Repressor of replication. So their repressor is similar or identical. So in that case, what will happen that the repressor of A is going to regulate the replication of B and vice versa, and that's where the interference comes. The A's repressor is going to interfere with B's replication and B's repressor is going to interfere with A's replication. And that's why what happens when these two plasmids start replicating, their repressors then interfere in their replication. And as a consequence, what is seen that one of the plasmid is lost from the host cell, and therefore they are called that they are not compatible or they are incompatible to live together. I will explain to you this aspect with an example. Okay, so let us see what is done in this experiment. Please follow this with me very carefully. Suppose you have a parent cell and you have these two plasmids in this A and B in this cell. So when this cell undergoes division, you will get two cells produced, and ideally you should get two daughter cells which should have A and B. Fine. But then what's happening here? When the cell is allowing to or preparing itself to grow, when it is preparing itself to divide, it starts growing in size. As I mentioned to you, the first preparation of the cell is increase in size. When the size increases, the repressor concentration goes down. When the repressor concentration goes down, then the plasmid replication takes place. So when the first replication is taking place, so there are two options to the cell. Either the B will replicate and you will get this situation, or A will replicate, you will get this. So 2 to 3 and 3 to 4, So before the cell splits into two, these two plasmids need to become four. Okay, so for that process, what is happening? First, this three plasmids are generated from 2 to 3. Okay, and this is the way. So either you will have two A's generated or two B's generated. In the second attempt, when this cell increases in size again, the repressor concentration goes down, then these three will become 4. Again, there are two options, that either A will replicate once again to give you a situation like this, or the B will replicate and you will get a situation like this. Means 2A, 2B, or 3A, 1B, like this. Similarly, this cell, which has 2A, 2B in the beginning, when it undergoes a second replication to make 4 plasmids, it is going to generate this is and this one, 3B, 1A. Now when this cell, which has now 4 plasmid, splits into 2, actual cell division takes place at this point. So to generate 2 daughter cells. So now you can see that this cell, when divides, it gives you 2 daughter cell, which will have one cell will have only A plasmid, and this will have 1A, 1B. Similarly, if this is the way, you see only A or B, or AB, AB. This is the possibility, one of the possibilities that both the cell will have the original plasmid pattern of A and P. Okay. And here you will see 1B. But the point here is whatever you may see, ultimately what you are seeing here, that as a consequence of this division, the cells are losing one of the plasmids. And when these cells undergo further multiplication, you know, the one of the plasmids will keep on will be missed out, and at the ultimately you will get the cells which will have either A or B, with very few cells having A and B. And this is what happens in plasmid incompatibility. So this incompatibility, it can also be explained based on this repressor hypothesis or theory. Okay, that it interferes. I mean, because they share or their repressor is identical or similar enough to interfere with each other's replication, and that is the reason why the two plasmids cannot coexist or cannot be maintained generation after generation. Okay, so with this, I conclude today's point of discussion, and what I have tried to explain to you is two features of plasmid biology: that how plasmids maintain themselves or maintain their number in a cell generation after generation, and also what is responsible for plasmid incompatibility. So thank you very much, and we meet again.